And I want to thank you for accepting my abstract I sent to, to the workshop. Okay, so, to the workshop. Okay, so well, to give me the opportunity, okay, to talk about zooplankton from this part of this world. Okay, we've been working on zooplankton ecology in the coastal upwelling system of Chile in the last 20 years, let's say, and trying to cover several aspects of zooplankton ecology from population, ecophysiology, metabolism, and community ecology. In this opportunity, I will concentrate just on one of the aspects which has to do with the interaction between zooplankton in the coastal upwelling system and this shallow oxygen minimum zone, which is a key feature in the coastal area of Chile. Okay, so what is the main issue? We know that from recent evidence that the oxygen deficient water are expanding worldwide. Okay, especially in some regions of the world, in particular in eastern boundary current system. Okay, and we don't know much about the biogeochemical consequences upon expansion of this low oxygen water. Okay, region with oxygen minimum zone, like the Humboldt current, yeah, should be considered as natural laboratories to do experiments to test the effect of hypoxia or low oxygen. And even in this region, we have a combination between low oxygen and low pH, which is another interesting issue. However, in the last few years, we've been working mainly with hypoxia and the effect of hypoxia on zooplankton. Okay, as I said before, the Humboldt current contains one of the most intense and extensive subsurface oxygen minimum zone. Okay, so let's talk about some of the general characteristics of this region, the Humboldt current. Okay, coastal upwelling is the key process controlling biological production at all levels. Okay, there's a strong variability over all time scale, like it happens everywhere. However, in this place, the most important variability has to do with interannual variability, which is related to El Niño southern oscillation variability. There is also important decadal variability, a multi-decadal variability, and all this interaction between different scale of variability can mask climatic change. So it's difficult to trace all the trends in, for example, in oceanographic variables. Okay. Another important characteristic, it has to do with the extension of the system. Okay, there's a long extension over the meridional gradient, and this creates a strong gradient in several oceanographic characteristics, like there are a strong gradient in temperature, gases, nutrients, and certainly in biological components. Okay. What about the distribution of the oxygen minimum zone? This is mainly controlled by upwelling variability. So, because the oxygen minimum zone is near the coast, when upwelling becomes more intense, the upper layer goes up. Okay, and if upwelling becomes depressed, then the oxygen minimum zone goes down deeper in the water. So this is a key feature, because if we want to characterize the distribution of the oxygen minimum zone and its variability, we have to deal with upwelling variability. Okay? In the context of climate change, we know that global warming is affecting the whole marine ecosystem. There is strong evidence for that. There's many work from different regions of the world that are showing that the increase in heat in the ocean is an important issue in the last 50 or 100 years. Okay, and that is related with the greenhouse gases. So what happened with the coastal upwelling system in terms of climate change? Okay, at the beginning we thought that because of global warming, we should expect in a coastal upwelling system a more stratification, okay, a lower nutrient input, and a system that is normally controlled by new production will become controlled by regenerated production, so the nutrient input and the carbon flux will become lower, okay? That was our expectation at the beginning. However, there is evidence that is showing that because of global warming, the wind, the southerly wind, will increase because of the difference between the temperature in the continent and in the temperature in the ocean. There's a greater difference, thermal difference, and that will strengthen the wind from the south to the north, and that will result in increasing upwelling, okay? And there is some records, long-term records, that are indeed showing an increase in the upwelling intensity in the last 50, 60 years, okay? So more upwelling implies colder water and apparently more nutrient input. The opposite what we were thinking at the beginning is that we have more upwelling, we have colder system just because of global warming. Okay? We also have some recent evidence now in the coastal upwelling, the Humboldt current, that indeed this area is becoming colder. This is the analysis of 20 years of data, and indeed it's showing a negative anomaly in temperature in the coastal area. Okay, and it happens the same in the Benguela system west of Africa. Okay, so there's a negative trend temperature. Then we have a system with colder and fresher and water. Also because we are getting more sub-Antarctic water, which are with less salty water. So salinity should become lower and colder in the upwelling zone of Chile. Okay. In our time series we have here of Concepción, we have a fixed station. We've been sampling in the last 10 years. Okay. What we have seen here is a seasonal upwelling, strongly seasonal upwelling, but it's hard to find clear patterns of internal variability, and even harder to find a trend. Yeah, it's just 10 years, so we may need more years to see any signal from climate change or from upwelling intensity. However, if we look carefully with the data, what we did here, we took some seasonal data from the beginning of the time series in 2 years, and at the end of the time series, after 10 years, we took another set of seasonal data, and we compare some variable, temperature, salinity, and density. And there is a significant decrease in the water column for the complete water column, okay? There's a significant decrease in temperature, okay? And there is also a significant increase in salinity in the upper layer. This because just because this is the evidence for more upwelling, because when we have more upwelling, we have colder system, but salinity increases because it's a subsurface water coming up to the upper layer, okay? Certainly also density will increase in the water column as well. So just by looking at this variable, we see that in a 10-year window, we are able to detect these changes in the upwelling intensity. Okay. Now, if we took more data, this is from the northern area of Chile, 30 years of data covering an extensive spatial area. What we took from three locations, we estimated the depth of the upper boundary of the oxygen minimum zone and the depth of the mixing layer as the base of the thermocline. Okay? And as you see here, there's an increasing upwelling, which is rising the oxygen minimum zone in the last 30 years. And this is a clear and very significant trend in the upper layer of the oxygen minimum zone and also in the depth of the mixing layer, okay? So the question now for us was, is there any effect on extremely shallow oxygen minimum zone and narrow thermocline on functioning of the plankton system? Just because we work with plankton, specifically with zooplankton, we say how zooplankton can respond to these changes. Okay? We know that zooplankton are considered to be good sensors for climate change, okay? Because they have very short life cycle. They can respond very rapidly to changes, to environmental changes. Okay? There are some important parameters you can easily get from zooplankton, like diversity, abundance, biomass, size structure. So all those parameters from the zooplankton can be considered as sensor for environmental changes related to climate change. Okay? So we thought that they may respond to these changes in the depth of the oxygen minimum zone. Some of the hypotheses we're dealing with are these two, for example, and just because of a shallower oxygen minimum zone and a very narrow mixing layer, plankton will become more aggregated in the upper part of the ocean. Okay? This will enhance biological interaction. Okay? Why plankton becomes more aggregated? Because most of the zooplankton are aerobic. Okay? They need oxygen. They need normoxic condition. So when the oxygen minimum zone is too near the surface, plankton goes up and stay in the upper layer. Okay, so they will become more aggregated, and when they aggregate, they also aggregate with their food. So that our hypothesis that will stimulate like feeding processes, metabolism, and also all kinds of biological interaction. And a second hypothesis related to that has to do with an enhanced biological activity. For example, feeding upon more aggregation will increase metabolic rate of zooplankton and then accelerating the carbon flux and nutrient recycling for the complete system. Okay, they appear to be simple hypotheses, however, not easy to test. And we've been working on this framework in the last, I would say, more recently in the last couple of years, and we are doing some experiment. And what I will do now is to show you some of the preliminary results we have for this experiment, and there's still a lot of work to be done. So we might get some good suggestions from you as well. Okay, we've been working in two places, in the northern of Chile and in the central south of Chile, here in Concepción. In the north of Chile, we have upwelling almost all year round, intermittent upwelling, and here in Concepción, just during the spring and summer. Okay, we have some sampling station we have been working in the last 20 years, say. Yes, we have a monitoring station here, but we visit this station near 4 times a year to get samples and do experiment over there. Okay? And in Concepción, we have been working mainly on station 18 here on the continental shelf. We also get sample here, live animal, to do experiment. And our approaches are mainly based on microcosm experiment. Okay? One of the parameters we are working on lately is the zooplankton community respiration as a metabolic index. Okay? In this case, what we do, we work with the complete community of zooplankton, okay? Like a black box. We don't know, we are able to know what they are. However, as in terms of metabolic rate, just the whole community, okay? We are doing estimate of zooplankton community rate and also feeding process through microcosm experiment. And the way to measure metabolism is by measuring oxygen consumption under control condition in short-term incubation, okay? Community respiration and feeding, now we are including a variable which is the concentration of phytoplankton as an index of dilution or aggregation effect, okay? Let's say we have a dilution effect when the oxygen minimum zone goes down and the thermocline goes down. Then we have a mixing layer that is deeper. So then we have a dilution effect. When the thermocline goes up, then we have an aggregation effect. And we can simulate all that in microcosm experiment by doing dilution and concentrating phytoplankton as a source of food for mesozooplankton. Okay. So some of the result I will show you here. Okay. Yeah. Okay. First, on this graph, we have a summary of the experiment or the estimate of zooplankton community rate at different times of the year. This data include both places, so there is not a distinction of the regional effect here. Okay, as you see here, there is apparently there's not much variability or seasonal variability. All these estimates have been standardized from carbon. They are carbon specific rate. Okay. At one time, we found a lot of variability. Each point here represents more than one experiment. We replicate, okay? And what about, we are also measuring at the same time the chlorophyll concentration as an index of food, yeah, availability. And certainly, if chlorophyll increases through the year and is higher during the summer, the spring and summer condition, okay? So from this data, what we can say, they say, well, first, community respiration appears stable through the year, okay? While chlorophyll is more variable, and there is no apparent correlation between food and community respiration, okay? On the other hand, when we do experiment with feeding, this is also a summary of all the experiment we have now. There is a strong functional response of zooplankton feeding to food aggregation dilution. So this is the dilution treatment, and there is the feeding here. Okay, so the more concentrated the food, the higher the grazing rate, which is the functional response, and that is expected, okay? Now, if we look at the single experiment, at the beginning we got very enthusiastic about this result because what is this suggesting here is that the more aggregated the food is, the higher the community respiration will be. See, like having more food means increasing the feeding rate, and that will result in an increased metabolic rate, okay? And that's may suggest that just by increasing metabolism, that the flux is getting higher as well, okay? So that's very motivating. However, when we look at all the data, incorporating the complete variability, things are not easy to, to interpret, okay? Here, what we have is the food concentration, and on this axis we have the community respiration, okay? Look at all this. There's apparently no relationship, okay? And however, here we have to consider there is a time source of variability, that is a local source of variability. So we have to look more carefully on this data, and we may need more data to see if that relationship can be sustained or not, okay? But anyway, it seems that, for example, when taking the mean value here, if we do not consider this experiment, because this experiment shows a strong variation, too much variation, okay? Perhaps there's a problem here. If we just consider all this experiment, there seems to be a positive relationship, okay? Yeah.
Just a small thing. It's you use natural assemblages of phytoplankton when you say
yes, yes.
Or culture?
No, natural assemblage.
Natural, you added a lot of different variables.
Yes.
So the type of phytoplankton, if they contain more or less nitrogen as well, if they have more spine or not, they can, so people can feel it or not. So, yeah.
Yeah. However, yeah, you're right. Yeah, fully right. However, chlorophyll is just an index of food resource, okay? Yeah, because then, for example, in the zooplankton community, you have organisms that feed on diatoms, on flagellate, on everything, okay? So in this case, chlorophyll, what it does is acting as a food index. Okay, yeah. Okay, another important issue here, it has to do with the community structure, and I think that's the next step. Okay, in both cases, for the prey and the predator. Okay, so that might make a difference. Okay. Now, a second question here has to do with, is a favorable environment just increasing upwelling? Is it good for the ecosystem? Someone might say, well, more upwelling, more nutrient, more primary production, more fish. Okay, great. Yeah. Now, I came back to the original data from the North Chile just to show that this is really impressive. The way that the oxygen minimum zone is going up. Actually, in the last few years, we've seen a lot of fish stranding and mass mortalities of fish and mollusks, and perhaps has to do with hypoxia or anoxia, okay, event. Okay. The trend is highly significant, yeah, in the rising of the oxygen minimum zone. Okay. So, is it good? Yeah. Well, looking at zooplankton biomass, and also lately we've been trying to look at the fish biomass as well. Okay. These are the 20-year, 20-year data of Northern Chile, not 30 years actually. 20-year data of Northern Chile with zooplankton biomass, and there's a significant negative trend, actually. Okay. Of Concepción, here in this coastal upwelling system, we have only 10-year data. However, we are already finding a significant negative trend. It's weaker, okay, than in the north, but still significant negative trend in biomass. What about fish? We don't know yet. After a strong El Niño in the early 80s, fish biomass from North Chile and south of Peru, yeah, in the upwelling system was increasing the biomass. Then we have a strong decrease during El Niño, during El Niño 1997, 98. Then an increase again. But in the last, let's say, 10 years, it's again a negative trend, okay? We are now working with more data. However, if this is a bottom-up control system, then you should expect lower fish biomass if zooplankton is becoming low as well, okay? No, I haven't. I do have the chlorophyll. No, there is no chlorophyll. Does not show any trend. Okay, not positive, no negative, no trend at all for so far. Okay. Well, just to summarize all these ideas. Well, the Humboldt current is becoming a cold spot just because of global warming. More upwelling is taking place in the coastal system. The implications are more hypoxia. The plankton may become more aggregated. Okay? The dilution aggregation of plankton can increase or decrease feeding and hence increase carbon flux. This is actually, we haven't confirmed this, just dealing with that. Okay? Increased feeding may not influence zooplankton metabolism, or I need more study to get a conclusion on that. Yeah. However, one of the suggestions we have is that increased fluxes of carbon and nutrient can result in lower biomass, in higher trophic levels, just because of faster turnover rate. That's one of the potential explanations on why zooplankton biomass is decreasing, okay? Because otherwise, we have no explanation. Chlorophyll, as we have said, does not have any trend, okay? So if we do have primary production stable or even increasing primary production, why we don't get more zooplankton, okay? And one of the possibilities is this one, that faster turnover rate implies lower accumulation of biomass, okay? And that might limit fish production, perhaps, okay? So, no good news for fish production, huh? Okay. Well, that's it. Thank you very much. Okay.